Hi, this is David Nguyen, co-founder of Brain Technology and inventor of the LCPC transform. In today's presentation, I'm going to show you how we can measure shape in a way that is distinct from area in 2D culture using the LCPC transform. In this presentation, we'll talk about two pilot studies proving our point. The first pilot study is from this paper right here that studied rat lung epithelial cells. In this study, the authors investigated autophagy and separated it from apoptosis. They treated cells with cobalt chloride to induce hypoxia, which also induces autophagy. But then they also co-treated cells with 3-methyladenine, which inhibits autophagy, but not apoptosis. But first, let's talk about how the LCPC transform works. Each cell needs to be segmented in order to obtain its outer edge. Then we overlay a parallel grid system, all of which extend from a baseline that's a uniform distance from the edge of every object so that we're consistent across all objects. This allows us to calculate the intersection of each grid line with the edges of the object, as can be seen in step 3. We can then calculate the distance of every intersection to the baseline. And for every radial line that connects to the object more than once, all distances on that line are summed together to get one value. Thus, we're able to plot the line position versus the sum distance to obtain the graph in step 4. At this point, we then do the fast Fourier transform on step 4 to obtain a frequency plot in step 5. And this frequency plot is a multi-dimensional representation of the shape that you began with. Furthermore, you can turn this frequency plot into one number that is a scalar value by summing all the bins together. Thus, when you sum all the bins together, you get one number that represents the shape you began with. This method in step 6 of summing all bins is what we did in this presentation. But first, when studying a heterogeneous system, such as particles or cells, right, in this 2D culture, in which the cells comes in different shapes and sizes, it's often helpful to calculate the quartiles of the distribution of the cell shapes such that you can compare the same quartile across different groups. And this often yields more useful information than comparing all cells collectively in bulk. For example, quartile 4 represents the largest particles, while quartile 1 represents the smallest particles. And quartiles 2 and 3 represent the mid-sized shapes that are often hardest for the naked eye to distinguish. We first looked at the cobalt chloride time course, and when we measure shape and area, we see that both capture very similar patterns across all 4 quartiles. Thus, nothing new here. However, when we measure shape in the double treatment time course, we found something very interesting. In the double treatment time course, we find a threshold effect when we measure shape via the LCPC transform. That is, after day 3, the shape index increases, and this increase is observed in all 4 quartiles. I'd like to point out the first insight we derived from this data. That is this tail down here. Now, quartile 1 represents the smallest cells, and therefore this tail here in this yellow box represents the smallest of the smallest cells, so to speak. In this study, the authors showed that when you treat with methyladenine, it blocks autophagy, which increases apoptosis. And so this makes sense because apoptotic cells shrivel, and therefore the size and shape of the cell decreases. Thus, this tail down here captures the increase in apoptosis that was observed. But an even better insight from this data comes from quartiles 4 and 3, because these quartiles indicate that there is an apoptosis-resistant subpopulation within these cells. And we can derive this because the authors show that when you treat with cobalt chloride, that induces hypoxia and autophagy, but when you then also treat with methyladenine, it blocks autophagy and increases apoptosis. So despite increasing apoptosis, there's a large number of cells that are actually increasing in shape, meaning they're doing the opposite of what apoptosis would do to a cell. Therefore, our shape analysis revealed that there is likely a subpopulation of cells that's either pre-existent in the cell line or induced by double treatment. This is the kind of insight that the LCPC transform can provide for your research question. And as a control, we did the same analysis, but measuring area to show that what shape measures is different than what area measures. As opposed to measuring shape, measuring area does not reveal the threshold effect between days 3 and day 5. This shows that shape and area are not the same things. Moving on to our second pilot project, we analyzed image from a study on retinal pigmented epithelial cells from chick embryos. But for this study, we intentionally used the radial grid system to show that there is an alternative to the parallel grid system. For the radial grid system, you calculate the centroid of your object, and then you overlay a radial grid, 360 degrees, spaced every 5 degrees. This allows you to calculate intersections between the radial lines and the edge of your object. You can also calculate the distance of every intersection to the center. If you linearize it, you see the graph in step 4. Then you apply the fast Fourier transform as before. So in this study by Ban et al., they extracted retinal pigmented epithelia from embryonic chicks at days 7, 10, and 14, and they either treated the cells with regular SF2 medium or medium that was conditioned from day 14. Examining their control group, when we measured shape and area and separate them into quartiles, we see that there are some similarities, as in quartile 4, you see an increasing trend across the 3 time points. But in quartile 3, you start to see a different progression. Shape shows a linear increase, while area shows a logarithmic increase. While in quartile 2, you see what looks like a logarithmic increase for shape, but for quartile, it's a parabolic progression. And then in step 1, shape shows a step increase, while area shows a linear increase with a very noisy middle group. Now, data like this may seem complicated and cumbersome, but it's actually very helpful if you're doing gene expression analysis by microarray or RNA sequencing. And the reason is because distinct progression patterns give insight into interpreting gene expression data. Here are 2 example scenarios. Cell shape is affected by cell membrane junctions, while cell area may not be affected by cell membrane junctions. Also, apoptotic or senescent cells have distinct shapes, even if the area of these cells have not changed much. Take, for example, quartile 1 of shape. The gene expression pattern might show that there is a strong apoptosis signature in the blue and orange groups and none in the gray. But if you measure only area, you would have a hard time making sense of why the first 2 groups would have a strong apoptotic signature. But if you measured shape in addition to area, as we've done here, you will realize that this actually explains that gene expression signature. Because quartile 1 are the smallest of the shape indices, the smallest cells, which are exactly the cells that you would expect to contain the category of apoptotic cells. So my point is, when you separate your data into quartiles and you measure both shape and area, you have more data at your disposal to make sense of gene expression data that might not be obvious through traditional measures such as area. Moving on to the conditioned medium group. So the authors took conditioned media from day 14 and fed it to days 10 and 7. And when we measure shape and area, they both picked up a similar program across all 4 quartiles of cell shapes. And this tells us that shape and area exhibit comparable changes, meaning conditioned medium is driving a very similar program across all 3 time points. And we can be confident of this because if you compare it to the control group, each quartile had a different progression pattern. When they were cultured in the standard medium and allowed to do autocrine signaling by secreting their own cocktail of proteins into the medium. To conclude, I want to reiterate my main point, that is, shape and area are not the same things. At brain scanology, we can measure shape without measuring area or volume. And what we find across the board is that shape provides much cleaner data than does area or volume. And on the point of gene expression analysis, I wrote a handbook called Tips for Interpreting Gene or Protein Interaction Networks. It is available on Kindle, but if you want a free copy, feel free to contact us at info@brainscanology.com. The problem of having gene expression data that comes from two treatment groups that look the same phenotypically to the naked eye is very common. And shape analysis, as I've shown you in this presentation, is one way of correlating phenotype with changes in gene expression. For more information, feel free to visit our website, brainscanology.com. For consulting services and software licensing, feel free to reach out at info@brainscanology.com.